faithfully conveying value, interpreting great books from around the world. Hello, this is Sally with Book Talks. Today we're exploring the book Between Life and Non-Life. Its author, Shinichi Fukuoka, was a researcher at Rockefeller University and Harvard Medical School in the US, and is now a professor at Aoyama Gakuin University in Japan. Alongside his molecular and cell biology work, he often publishes popular science articles. After its publication, Between Life and Non-Life received unanimous praise from major Japanese media like Asahi Shimbun and Yomiuri Shimbun, and was recommended by renowned scholars including Nobel chemistry laureate Yuan-sei Lee. The core idea of this book is a very profound question. Fukuoka says when you're walking on the beach, you'll see many scattered stones and shells. Oh, which one is living and which is non-living? You can probably tell at a glance, but what's the essential difference between living and non-living things? I is it really as simple as whether it breathes or not? This book revolves around narrating and discussing the essence of life. The scientific community has had extensive discussions on the essence of life. For example, in the 17th century, philosopher Descartes thought life was just a machine. This view influenced generations. In 1953, biologists Watson and Crick proposed the double helix theory of DNA. After that, people thought life was a self-replicating system, but Fukuoka doesn't agree with that. Uh, um, I just thought I didn't, and he believes that living organisms are actually a dynamic equilibrium system. To help readers grasp this, Fukuoka gives a vivid example. Imagine a delicate sandcastle on the beach that faces daily erosion from the sea. If no one repairs it, the sandcastle will quickly be washed away, vanishing without a trace. To maintain the sandcastle's shape, picture countless tireless spirits in the sea, constantly using new sand grains to fix damaged spots. Moreover, these spirits don't just passively repair the sandcastle, they actively knock down parts that might collapse, fixing them before the sea washes them away. It's through the efforts of these invisible to the eye spirits that the sandcastle can endure wind and waves and stand firm on the beach. Even if you observe for a long time, you won't notice any subtle changes. But actually, the sandcastle itself, even every grain of sand has been replaced. The sandcastle surface seems balanced, but essentially it's dynamic. That's the dynamic equilibrium Fukuoka emphasizes. Dynamic equilibrium is the essential property that distinguishes living from non-living things. All living organisms are in a state of dynamic equilibrium. The concept of dynamic equilibrium wasn't originated by Fukuoka. The first scholar who proposed the idea of dynamic equilibrium was Schonheimer, a German-born biochemist. In the 1930s, he designed a series of ingenious experiments using isotope tracing technology to show that food eaten by mice gets rapidly distributed throughout their bodies, assembled into new proteins and lipids, while old components are broken down and excreted, thus keeping their weight relatively constant. Mice can replace half the proteins in their bodies within 3 days, yet overall, there seems to be no change. This is just like the sandcastle on the beach we mentioned earlier. We seem to see a solid mouse, but in reality, what we see is just a river of flowing elements. Humans experience the same phenomenon. Our bodies are merely denser precipitates, with contents constantly renewing in various flows. As long as substances inside us keep renewing or are in dynamic equilibrium, it means we're alive. Mm-hmm. Mm-hmm. If the human body can't intake molecules from outside and only loses them, it loses dynamic equilibrium. And when people diet, intake of new molecules decreases, while outflow of old ones increases, which also means losing dynamic equilibrium. If this continues, it can lead to life-threatening dangers until turning non-living. In this new view of life, Self-replication isn't the essential characteristic of life. Dynamic equilibrium is. So when we see a seashell, the first thing we think of isn't self-replication, but the natural beauty of its order. Not only are they exquisitely structured, but they also grow continuously. It's a landscape from ceaseless atomic movement, a result of dynamic equilibrium. The great physicist Schrödinger also proposed a theory of life's dynamic equilibrium from another angle. In 1944, Schrödinger published a small book titled What is Life? He introduced the principle of entropy increase into life systems. The principle of entropy increase means that disorder in an isolated system always increases and never decreases spontaneously, like how a room always gets messier. Schrödinger believes that the principle of entropy increase applies to all physical phenomena. Life can't escape its grip. Genes and proteins lose function due to changes. Organisms cope by proactively removing chaotic parts and rapidly building new components to replace broken ones. It's through this approach that organisms expel accumulating entropy and maintain their stability. Schrodinger's view aligns with dynamic equilibrium theory. Both point to the same conclusion. The system of life is a dynamic equilibrium. After proposing dynamic equilibrium theory, Fukuoka argued it through three levels. First, the protein level. Second, the cell membrane level. Third, the genetic level. Oh, let's first discuss dynamic equilibrium at the protein level. Proteins are the primary agents of life activities. About 18% of human body mass is protein. How do they achieve dynamic equilibrium? To illustrate this, Fukuoka used a vivid analogy. He compared each protein to a small puzzle piece, but three-dimensional, not flat. The entire surface of proteins is covered with connectors, allowing complementary binding with others. These connections form vital body components like muscle tissue and molecular machines, such as ribosomes that manufacture proteins. For example, ribosomes, insulin, etc. With this puzzle mechanism, your body maintains orderly function. As proteins constantly degrade, but a continuous supply of new puzzle pieces instantly fills gaps, ensuring structural integrity. This raises a problem. If proteins bind too tightly, gaps become hard to fill because new puzzle pieces struggle to squeeze into correct positions. The wonder lies in protein puzzles not being rigid. They exist in semi-attached states, binding quickly, yet separating readily. Each puzzle piece is in motion, constantly testing all gaps and always fitting the most correct one. This binding ensures life's flexibility and complexity while maintaining macroscopic wholeness. Organisms continuously eliminate denatured proteins and promptly discard faulty ones through these mechanisms. Once protein replacement goes wrong and can't update in time, various diseases emerge, like Alzheimer's and mad cow disease. Both result from abnormal proteins that weren't promptly replaced. We stay healthy because faulty proteins always get replaced promptly. Only when errors accumulate beyond a threshold and the body loses self-purification ability does disease become unavoidable. Next issue. There are over 20,000 types of proteins in the human body, meaning over 20,000 puzzle pieces. With so many pieces, misfits are inevitable. So how do organisms prevent such errors? Fukuoka doesn't give a direct answer, but revisits Schrödinger's solution from his work. Back then, Schrödinger posed a strange question in What is Life? Why are atoms in our bodies so tiny? A single cell holds hundreds of thousands of atoms, and our bodies consist of countless cells, creating an extreme size ratio. What's the logic here? The atoms constantly undergo random thermal motion. How does this chaos build highly ordered life? Schrödinger proposed the square root law of errors. Faulty particles usually number the square root of total particles. For example, in 100 atoms, about 10 might malfunction. If 1/10 of an organism's atoms malfunction, it couldn't form ordered structures and would collapse. But in larger organisms, say with 1 million atoms, only about 1,000 might err. Compared to 1 million, this error ratio drops to 1,000. Meaning, the more atoms in an organism, the smaller the proportion of errors. When organisms grow large enough, error proportions become negligible, enabling stable existence. That's why atoms are tiny and organisms large. Only this way can sheer quantity counteract individual atomic errors. This principle also applies to proteins, the building blocks of humans. There are over 20,000 types, each in astounding numbers. Just actin or insulin proteins number in the hundreds of millions. So, the total protein count in the body is astronomical. When you take its square root, the error rate becomes extremely low. Even if some proteins land in wrong positions, they get quickly dismantled, disappearing into the flowing protein river, causing almost no impact on the organism. Next, let's discuss dynamic equilibrium at the second level. The cell membrane, what exactly does it do? Simply put, it's like a courtyard wall dividing the cell into inner and outer sections. This ensures stable internal conditions for orderly cellular activities. All cells have membranes. Pancreatic islet cells are no exception. Islet cells synthesize proteins like insulin and digestive enzymes. After synthesis, these must be transported out to function. So, how do proteins scale the wall? On the membrane's inner surface is the endoplasmic reticulum, constantly budding small membrane structures. Imagine the cell membrane as a balloon. These small structures are mini balloons budding inside. Protein transport works like this. A mini balloon wraps synthesized insulin in its belly, attaches to the main balloon, and their membranes fuse. Then the mini balloon opens outward, releasing insulin. These mini balloons are scientifically called vesicles, collectively forming the vesicular transport system within cells. You might wonder, why such an elaborate transport system? Simply opening a hole would destabilize the cell's interior, risking collapse. The vesicular system doesn't just export, it also imports materials from outside. Their channels flow incessantly. Vesicles move in and out, eventually becoming part of the cell membrane itself. This means the cell membrane is also in dynamic equilibrium. To study membrane formation, Fukuoka's team analyzed vesicles carrying digestive enzymes, discovering they all contain a special protein. They named this protein GPR. While studying GPR's gene sequence, Fukuoka found new evidence supporting dynamic equilibrium theory, leading to our final topic, genetic level dynamic equilibrium. Fukuoka's team wanted to know the specific function of GPR. They not only analyzed GPR's gene sequence, but also decided to delete the GPR gene from cells to see what would happen. The reason for this idea is that scientists once viewed life as a machine, like the densely packed, colorful circuit board behind a TV. If we want to determine what a part does, we remove it and see how the TV reacts, then deduce its function. For example, if the TV loses sound after removing a part, that part might relate to sound. This is simple logical thinking. This way of thinking is also applicable when studying cells. Fukuoka believed that if they deleted the GPR protein gene from cells, no GPR protein would be produced. And by seeing what happens to the organism, they could deduce GPR's function. After arduous exploration, Fukuoka's team finally obtained a mouse like this. All cells in this mouse lacked the GPR gene, so no GPR protein was produced. And then they waited for some miracle to happen to this mouse, but nothing occurred. The mouse appeared perfectly normal. Except for lacking the GPR gene, it had almost no differences from other mice, and its vital signs were unaffected. It was as if the gene deletion never happened to it, which amazed Fukuoka. It was incredible. It's like knocking a part off a TV's circuit board, yet the TV works perfectly fine. They first suspected an experimental error, So they hurriedly conducted a full analysis of the mouse. They found no errors. The gene was deleted, but the mouse was fine. Behavior, metabolism, chemical indicators, all were normal. How could this happen? After deep contemplation, Fukuoka suddenly understood. There's only one explanation for this. The GPR gene isn't indispensable. It can be functionally substituted by other genes. Substitute genes. After deleting the GPR gene, other genes immediately stepped up to perform tasks that should have been done by GPR. This is like a striker on a football field who's responsible for scoring goals. But when a striker gets sent off, a defender can still score goals. When these clues came together, Fukuoka realized life isn't a machine. Comparing life to a TV was a serious mistake, and gene deletion is entirely different from removing parts. Our life is actually a one-way process, where all changes strictly follow a timeline. At certain life stages, specific components become active. If something's deleted, the dynamic equilibrium system can create substitute components to minimize gaps and maintain operation. This is genetic-level dynamic equilibrium. Genes aren't static sequences, but information carriers that can activate, deactivate, or even modify functions. Life systems contain numerous movable equilibrium points. If an organism lacks an enzyme, seeming unbalanced, the dynamic system immediately opens alternative pathways to restore equilibrium. Hence, life systems have redundancies, multiple similar genes and reaction pathways ready for emergencies. The mouse developed normally despite a key gene deletion because the gap was promptly filled. If damage is severe beyond dynamic equilibrium's repair, death follows. The system only moves forward. Life can't reverse, every moment is irreversible. Facial hair grows but won't retract. Dead cells can't revive. Reckless external interference causes irreparable damage to dynamic equilibrium. Cracking an eggshell to inspect inside prevents the chick from ever hatching. Even seemingly minor issues are only manageable due to the system's flexibility. Compensating. Life-environment interaction is origami. Foldable, but not unfoldable. Once folded into a complex paper plane, restoring its original state for refolding is nearly impossible. So Fukuoka suggests, if asked what life is, say it's origami that can't be unfolded. This is the essential difference between seashells and stones, between living and non-living things. A small stone lacks dynamic equilibrium. It simply exists. But behind a seashell, countless stories of dynamic equilibrium unfold. Their collective actions uphold life's exquisite structure. Structure results from repeated origami folds, and through gene expression, this complex process repeats, folding relentlessly in one fixed direction. Interference only diverts it onto unintended paths. Or, as Fukuoka said, humans can't manipulate life like machines. We can only bow before nature's torrent, faithfully recording life's aspects. There's no other way. All right, that's our interpretation of between life and non-life. Reading notes are below the audio. See you tomorrow.